Recombinant DNA technology is an important aspect of biotechnology that involves the production of recombinant molecules, that is, where the DNA comes from two different sources. We have been continuing this series on recombinant DNA technology, and we have covered 4 lectures on talking about different types of cloning vectors. Now, when we talk about DNA from two different sources, these two DNAs, the one comprises a gene of interest, which we want to either sequence or amplify, that is produce more number of copies, or want to express it to that is we need its protein product, and the other DNA is generally a vehicle which carries this gene of interest into a different host organism where the downstream manipulations take place. So, we have talked about the cloning vectors. Today we will be talking about the DNA source from where the gene of interest or for different manipulations we use in the recombinant DNA technology. So, the particular learning objectives for this session would include to understand the fundamentals of isolation of total cell DNA, to understand the fundamentals of the isolation of plasmid DNA, to understand about the visualization of DNA, to understand the quantitation of DNA. So, basically the source of DNA from which we isolate the gene of interest, how do we isolate that? So, for that concept, again, when we isolate it, we can isolate it either from a bacterial cell or a prokaryotic cell, or we can isolate the DNA, a gene of interest could be lying in a plant cell or an animal cell or bacteriophages. Particularly, cloning vectors are also DNA. So, we need to isolate them too, because we are doing in vitro manipulations for the recombinant DNA technology. We need to isolate these DNAs and then produce a recombinant DNA molecule outside the cell, outside the or in the vitro. So, these DNA we will talk about that how these DNAs are isolated and purified. So, the references used for this particularly for the educational purposes is primarily T. A. Brown. Most of the figures come from T. A. Brown, and another one, the Advanced Molecular Biology and Concise Reference by Twyman. So, otherwise stated, the references are these basically 2 textbooks of molecular biologies. So, we start with the preparation of total cell DNA. Now, this total cell DNA, as I have already told, is often required as a source of material from which to obtain the genes to be cloned. So, we have looked at the recombinant DNA technology as an important process of molecular cloning. Cloning basically means producing more number of copies. So, if of any gene we want to produce more number of copies, we tend to take that gene of interest, clone it into a cloning vector which act as a vehicle to introduce it into the host cell, and then in the host cell this cloning vector replicates, and along with it the genes replicate, the gene of interest replicates and produce more and more number of copies. Now, as I said, this can be done for production of proteins, or this can be done for any other purposes or the downstream applications which we will take up in subsequent sessions. So, this source material basically is the DNA and comes from any of the cells, the bacterial cell, the plant cell, or the animal cells, or even the bacteriophages that we take up. Now, this total cell DNA, when we talk about it, consists of the genomic DNA or the chromosomal DNA of the organism, along with any other additional DNA molecules such as, for example, plasmids. So, we have seen that the plasmids can be used as the cloning vectors. So, generally in a cell there is a chromosomal DNA or the genomic DNA that is present, and apart from that there can be additional DNA molecules primarily which are called as the plasmids. These are the episomal or the extra chromosomal content which is there. So, the total cell DNA, when we need to prepare, we basically prepare the entire DNA preparation and purify the entire thing, and the plasmid will also come with it. So, we generally isolate this for taking out the genes or the gene of interest that need to be cloned. Now, this may be the DNA from a culture of bacteria, as I said, from a plant, from animal cells, or from any other type of organism that is being studied. Obviously, the mechanism is much simpler when we isolate the total cell DNA from the bacteria, and difficult when we do isolate from the plant cells or animal cells, but we will talk about the basic general mechanism, and then we will look at certain modifications which are done so as to isolate the DNA from the plant cells or animal cells or the bacteriophages which are there. Now, the procedure for total DNA preparation from a culture of bacterial cells can be divided into 4 stages. Now, basically you have to understand this fact is that to start with the recombinant DNA technology process, because you need the DNA as the source, you need to isolate this DNA from some cells. Now, whatever these cells are, there are 4 stages how you can purify this total DNA from those cells, and those stages are, the first one is that a culture of bacteria, we will look primarily at the bacterial preparation of total cell DNA, and then look at certain modifications which are required for the plant cells or higher organism cells. So, a culture of bacteria is required basically, which is grown and then harvested. So, there are 2 terms used here. One is that a culture of bacteria is grown and then it is harvested. Now, to grow a bacteria, so we have single cell bacteria, we can have single cell bacteria, or we have a pure culture of bacteria. So, that bacteria will act either as the host organism to which we add a recombinant DNA molecule, or that bacteria act as the source for the gene of interest as well as source for the cloning vectors. So, because the source of DNA, both the sources for recombinant DNA technology are DNA, and the recombinant DNA molecule is also DNA. So, the whatever purpose is there, you basically need to understand the protocol for the isolation of total cell DNA from these cells. So, first of all, these cells need to be grown in large amount of numbers. So, the bacterial cells need to be grown and then they need to be collected so that the DNA can be isolated from there, from these cells. Now, growing the bacterial culture basically requires certain growth media. Now, growth media is like the food for growing bacteria. It is the medium that must provide a balanced mixture of the essential nutrients at concentrations that will allow the bacteria to grow and divide efficiently. Now, we understand that bacteria is ubiquitous. It is everywhere. It is growing in aerobic conditions, anaerobic conditions, in extreme environments. You can find bacteria or microbes everywhere where you can see. So, it is lying in your gut, it is lying in the environment, it is lying in the thermophilic environments, it is everywhere. So, there they have certain natural resources which they are utilizing for their growth. Now, when we want to grow this bacteria under controlled lab conditions, we need to provide certain essential nutrients and resources so that it can divide efficiently. So, division here means, or the growth in case of for example, bacterial culture means increase in the number and not just increase in size. When we refer to growth in case of human beings, we generally tend to talk about the growth in size, that from a baby to an adult, that is the growth. But in terms of bacterial or microbial world, the growth basically refers to the increase in number or the division of the cells to produce the progenies. So, the medium which is there, the growth media, because we are doing it in the lab conditions, must provide a balanced mixture of the essential nutrients. Now, there is a whole diversity of bacteria that is present. Depending upon what kind of bacteria we want to utilize, there are n number of formulations which are there for growth media, which are prevalent. So, they have been standardized, they have been improved upon, so that the whatever bacteria we want to grow, it grows very efficiently and divides very efficiently and gives us a higher cell number in lesser amount of time. So, these media are there is a classification of growth media. There are simple media, there are complex media, there are defined media, undefined media, selective media, variety of ways in which these growth media can be classified. I will just touch upon some of the aspects here, because we just need to grow the culture of bacteria. And in generally, the recombinant DNA technology, we use E. coli as the most common, Escherichia coli, the gram-negative bacteria, as the most common bacterial host. So for growing that thing, we require very simple, very defined media which is there. So the defined media basically refers to the fact that the nutrients are exactly known. Undefined media refers to the fact that you do not know the exact nutrients which are present. For example, you use yeast extract. Now, yeast extract has multiple nutrients which are available, but yeast extract as such is a mixture of all those nutrients. So, when you add yeast extract, it becomes an undefined media, because we do not exactly know the composition of yeast extract, but we know that this yeast extract enhances or stabilizes the growth of certain kind of microbes. So that kind of a media, then it also becomes a complex media, because not the simple nutrients are used. So it becomes a complex media. Simple media is where very simple ingredients are used which are readily available, and they are very much defined that these things are to be added in such a quantity. Then we have something which is called as a minimal media and a rich media. So minimal media is a media which contains very basic nutrients which are essential for its survival. So when we want to study the growth of a particular bacteria under the influence of any exogenous agents which we are adding, be it the chemicals or be it radiations or be it any other molecules which are prevalent, if we want to plan a study like that, we use a media which is a minimal media, because the minimal media will just make sure that the bacteria grows, and it does not require the bacteria to be dependent or to alter the bacterial growth, because it is having a very rich source of nutrients. So it is just growing normally in the minimal media, and therefore whatever effect is produced by the addition of any kind of an exogenous agent which we want to study. So rich media are something which have like sugar and very complex media which is actually affecting the growth of the bacteria, which is enhancing the bacterial growth also. So again, depending upon our experimental plan, depending upon what we want to do, we choose a growth media for the growth of the bacterial host. In here, we are talking about the growth media just for the culture to have the number of cells from which the DNA can be isolated. So the basic thing here is that the bacteria should be able to grow and divide efficiently, because we need a large number of cells. Large number of cells will ensure a larger amount of DNA that can be isolated from it. And then generally the growth curve is also standardized. So when we take this culture and isolate the DNA, we generally do not take the cultures in the initial lag phase or initial exponential phase, but the late exponential phase where the number has reached too much. If it enters the stationary phase or it goes into the death phase, then also we do not get very large quantity of DNA. So the growth curve of the bacteria is also very important, so that one has an idea that at what time point the culture needs to be harvested so that large amount of DNA is produced. So here I just give you the constituents of two simple media. One is M9 medium and another is a Luria-Bertini media, LB media. Now this Luria-Bertini media is very much needed for the is very prevalent for use in the recombinant DNA technology, particularly for growing the E. coli host. So as you can see here, there is tryptone, there is yeast extract. So it's an undefined medium, and it is a complex media. But in the M9 medium, you can see the particular constituents actually are laid down, that whatever is required and in what quantity. So this become a kind of a defined media. Now media, again, when we are growing a particular bacteria, we can grow it in the liquid things. When we are growing the culture so as to collect the number of cells for the isolation of DNA, we grow it in a liquid medium, and the liquid medium is called as a liquid broth. But we can also maintain the culture on the solid media, where we add chemicals like agar into this medium, and then when we pour it into the Petri plates, it becomes solidified, and therefore the culture can be grown onto it by spreading and streaking methods, and can be maintained for some time, or they can be subcultured again and again for continuous use in the laboratory. For the isolation of DNA and the purification of DNA, however, we inoculate the cells into the liquid broth containing any of the media, depending upon what kind of downstream manipulation we need to do. So, the growing is very important, the growth of the culture is very important, the stage of the growing culture is very important for good amount of DNA to be isolated. The next thing is, when we grow this culture in the bacteria, depending upon the optimum growth temperature, depending upon the optimum time does it take, after that the cells are harvested. Harvested means that the cells are collected and rest of the media is just thrown out. So, for that what we use is what is called as the centrifugation. So, because we need to isolate the DNA from the bacterial cell, therefore, the culture is grown and then put into tubes and then centrifuged or rotated at high speed, and at these high speed, due to the centrifugal force, the cells which are heavier tends to pellet down. So, we can decant the supernatant and then use these culture cells, the bacterial cells, to isolate and purify the DNA. So, the first and foremost step in the preparation of total DNA or plasmid DNA also is the growth and the harvesting of the cells from which the DNA need to be isolated. Moving on, the second step actually requires that the cells debris or the cells are removed, and what we get is the cell extract. So, the cell needs to be now opened up. The cell contains the bacteria. We have the number of cells now. Now, these cells need to be opened up. They need to be lysed, as it is called as. They need to be disrupted so that the cell extract can be prepared. So, the cell extract can be prepared by physical methods or chemical methods. The physical methods involve the cells that the cells are disrupted by what are called as the mechanical forces, such as a technique known as sonication. So, in sonication, high sound waves are produced and the cells are lysed in that. So, various mechanisms, these are the physical methods, or some kind of mechanical shearing is there. All those things will break open the cell to release the content into the solution. So, whatever cell pellet we got, we again resuspend into a smaller volume of a buffer, and that in that buffer we provide either the physical methods or the chemical methods. So, chemical methods involve the lysis, which is brought about by exposure to certain chemical agents that affect the integrity of the cell barriers. So, important aspect here is that we need to break open the cell wall and the cell membrane and want to release its content so that the DNA can come out. So, there are two mechanisms usually involved, the breaking up of the cell membrane and the cell wall which is there. So, that is stabilized by certain ions, that is stabilized by the osmotic pressure, etc. So, all these needs to be disrupted. So, if we add certain chemical agents, then this thing can be disrupted, and because of this disruption, the cell membrane, the cell wall can be broken, the cell membrane can open up, and all the contents of the cell will come out into the solution. So, for the chemical methods, as you can see, there are certain important chemicals such as the lysozyme. Now, lysozyme is an enzyme that is present in the egg white and in secretions such as the tears and saliva, and this digests the polymeric compound that give the cell wall its rigidity. So, for the cell to maintain its structure, the cell wall, there are compounds, there are pressures which are maintaining the cell structure. So, there are so all these things, as I said, needs to be disrupted so that the cell becomes liable to lysis, and then when we put the agents, it will lyse and it will release the DNA into the external environment. So, lysozyme act as an important chemical agent, as an important enzyme that is used to digest the cell wall, and it loses its rigidity. Other thing is what is called as the ethylene diamine tetraacetate, or EDTA as commonly called as. It removes the magnesium ions, the divalent cations that are essential for preserving the overall structure of the cell envelope, and it also inhibits the cellular enzymes that could degrade DNA. So, we also add, for chemical purification of total DNA, we add EDTA, and this EDTA chelates the divalent cations which are essential for preserving the sanctity of the cell envelope. So, the cell becomes more liable to break open. and when the cell break open, then the nucleases can also be released. Then these nucleases will again cause the sharing of the DNA, which we do not want. We want the pure and the intact DNA which is there. Therefore, this EDTA also acts to inhibit the cellular enzymes that could degrade the DNA. So, a combination or in isolation, lysozyme and EDTA are used to make the cell labile for the breakage. Then other things which are used is such as the detergent, for example, sodium dodecyl sulfate or SDS. It aids the process of lysis by removing the lipid molecules and thereby causing the disruption of the cell membranes. So, detergent, as you know, it tends to form the lipid micelles of the lipid bilayer, what are called as the spheroplast in plant cells. So, these SDS agents, they cause the membranes to break open, and by forming these kind of structures, they act to remove the lipid molecules, and finally the cell is broken or lysed and the content comes out. So, after this cell is lysed, now we have in this solution the cell debris, as well as we have the DNA and the RNA and other biomolecules, the proteins, the carbohydrates, which are all lying in the external environment now. So, to harvest now the biomolecule and particularly the DNA, which we want to purify and isolate, we again centrifuge this thing and we get the cell debris which is at the bottom, the membrane and the wall and all those organelle parts. They just settle down because they are heavier, and the solution now contains the mixture of the biomolecules, which is DNA, RNA, protein, or in other cases carbohydrates, etc., can now be found in the solution. So, this second step is again very important. So, we grow the culture, we harvest the culture, and then we lyse the culture so that the biomolecules are released into the external environment. Coming to the third stage is that the DNA is now purified from the cell extract. As we have seen, the cell extract contains other biomolecules along with the DNA. It contains RNA, it contains proteins, it contains carbohydrates. So, now what we need is the purification of DNA from this cell extract. Now, two approaches to DNA purification are usually followed. One is by treating the mixture with reagents which degrade the contaminants, leaving a pure solution of DNA, and another is by separating the mixture into different fractions, one of which is pure DNA. So, we will take one by one and just talk about one or two technique that is involved in it. So, there are n number of techniques depending upon the kind of DNA we are isolating, the cells we are using to isolate the DNA. But we just touch upon the basics of the some simple techniques so that you have a conceptual idea of how the total cell DNA or the plasmid cell DNA is purified and isolated and purified. So, the first is basically the treating the mixture with reagents that degrade the contaminants, leaving a pure solution of DNA. So, in this, one of the method is the organic extraction and the enzyme digestion. So, these are the two methods. So, one is the organic extraction. So, what do we mean by that? Now, we have a cell extract which contains a soup of biomolecules that has been released from the cell. So, what we do is we add the phenol, or 1:1 ratio of phenol is to chloroform to the solution and mix it well. Then when we centrifuge it or when we spin it, then the organic and the aqueous phase, they separate out. So, this is the aqueous layer that contains the DNA, and this is the organic solvent layer, which is basically the phenol. And as you can see, a band appears at the interface that will contain most of the proteins. This is a white colored band that appears, that contains most of the protein. The proteins coagulates and they form a band here, and some of the RNA molecules are also coming here. So, this now contains the most of the DNA and some of the RNA molecules. So, we have kind of coagulated and removed the protein by extracting it with the organic extraction. Now, this phenol extraction, one can do it just once if not too much of protein is there, but from certain cells which contains too much of proteins, repeated application of organic extraction needs to be done to get a purified extract of the DNA or the nucleic acids which is there. So, it can be done twice, and then a round of extraction which only chloroform can be done, and the chloroform removes the traces of the phenol which will be left out, because we carefully take out this upper upper layer and put it into another tube. So we might just contaminate it with some of the phenol that can come in here. So the last step of extraction we do with only chloroform, so that any traces of phenol is removed. Now these traces, why we need to purify DNA? You know, we need a very, very purified DNA because recombinant DNA technology or in vitro genetic manipulation involves various enzymes, and it is a very, very sensitive process. If the DNA is not purified, then we are not giving the optimum conditions for the downstream enzymes that we are using to manipulate the DNA molecules. So if we are not using that, then our downstream reactions will just not work. So we need purified DNA so that all our subsequent reactions will work, and these chemicals such as phenol or proteins or RNA will not hinder in the manipulation of the DNA molecules. So this is about the organic extraction, but no organic extraction also leaves behind some of the molecules such as the RNA molecules or other maybe some of the protein molecules may also remain. So there are other methods such as the enzyme digestion. So in this we use enzymes such as proteinase, the proteases such as the proteinase K or pronase, and ribonucleases such as RNase H. So these purified enzymes, can be used and put into this extract, and then again the DNA can be precipitated from that. We will talk about it later, but an application of these enzymes can remove most of the proteins and most of the RNA. It will denature or digest all the proteins and the RNAs to give a purified DNA in the supernatant which is there. So again, purification, this is very important method. More sensitive methods which are very prevalent are such as the ion exchange chromatography. So in ion exchange chromatography, the attachment of the DNA to an ion exchange particles are there. So these ion exchange particles are termed as resin, and as you can see, it is a reversible process. As we add the salt concentrations, the DNA will get removed from this resin molecules or the ion exchange particles. So because the DNA is negatively charged, we can use positively charged molecules as the resin, and we can attach the DNA by increasing the concentration of salt and adding into it the molecules which are attached to the resin will get dissociated. Now the sequence of dissociation will depend upon the concentration of the salt and the affinity of the molecules to the resin. So as we keep increasing the salt concentration, the more and more tightly bound molecules tend to come out and leave the resin which is there. So when the lower concentration of salt is there, loosely bound molecules will come out, and as we increase the higher salt concentrations, tightly bound molecules will come out. So because the DNA will be tightly bound, and we can differentiate, we can sequentially elute or unbound the samples from the resin by increasing or using different salt concentrations. So this ion exchange chromatography became an important aspect. We can just directly add the DNA molecule into the resin, or more important thing is to use such kind of columns. So or in a normal syringe, you can pack this syringe with the resin, and this resin is an ion exchange resin, and then you can put your solution onto it. So as you can see, the solution will gradually just be here, and then you use salt concentration. So when you use a lower salt concentration, the molecules which are loosely bound will come and can be collected. So when you use a higher salt concentration, then subsequently when you use higher salt concentration, the tightly bound molecules such as the DNA can also be eluted. So sequential elution of different molecules. So this will require the separation of the mixture, and therefore we will get one component DNA as the purified material which is there. So ion exchange chromatography, in this case we are using iron resin. Then there is another similar method that involves a compound which is called as guanidinium thiocyanate, which has two properties that makes it very useful for DNA purification. One is that it denatures and dissolves all biochemicals other than nucleic acids, and therefore can be used to release DNA from virtually any type of cell or tissue. So not only does it separate because of the binding affinity to the resin, it basically uses these are the silica particles which are used, and but it also denatures and dissolves all other biomolecules, so they can be easily isolated. Another property is that in the presence of guanidinium thiocyanate, the DNA binds very tightly to the silica particles, and it provides an easy way of recovering the DNA from the denatured mix of biochemicals. So similar mechanism we used by using the columns, and that column is packed by silica particles, and then we add the solution of a guanidinium thiocyanate, so that the silica particles in the presence of guanidinium, and when we add the cell extract, the DNA will bind tightly. So this guanidinium thiocyanate will digest all other biomolecules, the carbohydrates, the primarily proteins, RNA, etc., and the DNA will remain tightly bound to the silica. So when we keep adding a guanidinium thiocyanate, these other molecules will be digested and will be eluted out. However, the DNA will remain tightly bound to the silica particles. That can be removed by addition of water. So when we add water, the DNA will also be eluted, and therefore it can be separated from the all other biomolecules which are present. So these are some important mechanisms on how from the cell extract the DNA, the DNA phase or the DNA part can be purified. The last aspect comes is when we have to concentrate the DNA. So what we have done is we have grown and harvested the cell pellet. We have lysed the cell pellet so that all the contents come out. That in all the contents that have come out, we have purified the DNA portion. Now this DNA portion, when we try to bring it into a solution, it can be into a large volume. But what we require is a higher concentration of DNA. So higher concentration of DNA is basically when you start with a higher number of cells and a very growing stage of cells which is there. But in fact, even after that, you need to sometimes concentrate into a very smaller volume, so that for downstream manipulations you use a higher concentration of DNA. So you need to reduce the volume and concentrate the DNA. The basic mechanism to use it, the very generalized mechanism, is the ethanol precipitation. So in the presence of salt, strictly speaking, monovalent cation, so whatever after purification the solution we are getting, we add monovalent cations to it, such as sodium ion. So we add like some sodium chloride solution to it, and at a temperature of and then add 100% absolute alcohol and incubate it at -20 degrees Celsius for half an hour. So after the temperature of -20 incubation at -20 degrees Celsius, absolute ethanol efficiently precipitates the polymeric nucleic acids. So what we basically get is that the nucleic acids will be precipitated out. Now again, there can be two methods that is generally used. One, if there is a large amount of DNA which is there in the and there is a volume of DNA, but you still want to reduce the volume of DNA, but the amount of DNA is large. So what you can do is you can just you can add the salt solution and you can pour the ethanol on the top of it, and by using a glass rod. Now glass rod will kind of, there will be an electric charge difference, and therefore the DNA molecules will stick onto the glass rod. So this is called as the spooling method. So you can just swirl the glass rod inside this solution, and all the DNA will come onto the glass rod, which you can just take out and suspend it into a lesser amount of solution to make a concentrated DNA thing. So this is basically that absolute ethanol is layered on the top and the DNA is spooled out by using a glass rod. But if the solution is diluted and not, too much of DNA is not present, then what we basically do is, for less concentrated solution, ethanol is added at a ratio of 2.5 volumes of the absolute ethanol to 1 volume of DNA solution, and then precipitate it. Then we incubate it at -20 degrees Celsius for half an hour, or at higher temperatures for overnight, for 4 degrees Celsius at overnight, or even colder temperatures we can incubate if we have the facility at -80 degrees Celsius for efficient precipitation. and then we centrifuge it. So when we spin it again, the DNA that has precipitated out would pellet out. So we can discard this ethanol and then we can air dry this pellet and suspend it in the required volume to get a concentrated sample of the purified DNA. So this is the fourth stage of isolation of total DNA, which is basically the concentration step. Now, then we come on to the fact that we have isolated the total cell DNA. I will talk about the isolation of the plasmid DNA also, but before that I am talking about, like, if you when you want to use it for the experiments, so what kind of experiments we will be using. We will be discussing about the restriction and modification enzymes in our next session, which is basically that when I talk about making a recombinant DNA molecule, we are getting DNA from two different sources and we need to put them together. So now put them together, basically when we talked about cloning vectors, they are circular molecules. We need to cut them, we need to open them up, and then we used to put the gene of interest and then we need to ligate them again, so that a circular molecule is formed. So all these manipulation of digestion, ligation, introduction into the host, etc. requires certain techniques which are involved, require certain enzymes which are needed. So for putting up any in vitro reaction, we need to know the measurement, or we need to know the concentration of the DNA that we have isolated or purified, because when we talk about commercial enzymes that we will be using, then that commercial enzymes will come in terms of the as units of their activity. That 1 unit means that 1 mg of or 1 microgram of DNA will be digested in so and so manner. So for using, how much enzyme we will use, how much enzyme we will use to ligate it, how much DNA we use to introduce into the host. So for all these things we need to know the amount of the isolated purified DNA, and therefore there are techniques to measure the DNA concentration. DNA concentration, one of the very prevalent technique and very commonly used in laboratories and for recombinant DNA technology or any kind of biotechnology applications, is that DNA concentrations can be accurately measured by ultraviolet absorbance spectrophotometry. So the absorbance, spectrophotometry, where the light is absorbed, and so we will talk about the absorbance spectrophotometry, but the light will be in the range of ultraviolet radiations. So the amount of UV radiation which is absorbed by a solution of DNA is directly proportional to the amount of the DNA in the sample. So we will just check how much amount of DNA is present by taking the known concentrations of DNA. So we already know the concentration, we can use the commercial DNA for that. So commercial DNA will come with a specified quantification that so much amount of DNA is there, we can use to make serial dilutions, and then we can make different concentrations, and that different concentration we can check the absorbance at the UV, which is the maximum absorbance is at 260. So we can measure the 260 nanometer, the absorbance of DNA, and we can create a standard graph, and then for unknown DNA concentration we can extrapolate it and find it. We can do it manually. There are instruments which do it by itself. Then spectrophotometers require a little larger volume of DNA, but now there are many such facilities or many such equipments which are there, such as nanodrop, which requires just 1 microliter of DNA or even less than that to find the concentration of DNA. So whatever you have isolated, you are not wasting just to find the concentration. You use very less amount, and then there are softwares which are attached to the machine which will directly calculate and tell you the concentration. So a standard graph is already incorporated in it, they are calibrated and everything is there. All you need to do is you just put the DNA drop, a very, very tiny drop in between the two electrodes, and when the radiation is passed through that, in between those two panels, it can just tell you the concentration. So, and the range is in nanograms, it can basically goes and tells the concentration of DNA in a particular sample. So, as I said, the absorbance is usually measured at 260 nanometer, and the maximum absorbance of 260 nanometer of 1 corresponds to 50 mg of double-stranded DNA per ml. So this has been found out that the absorbance of 1 of the solution will correspond to 50 mg. So anyway, we can just correlate and find out the concentration of the double stranded DNA per ml in any of the sample in which we have isolated the DNA. Apart from just telling the concentration of DNA, this UV spectrophotometry also can be used to check the purity of a DNA preparation. So we have seen that during the isolation and preparation of total DNA, we have employed the steps in which we tried to purify the DNA and remove the content of proteins and RNA, etc. And but still, there can be the possibility that these molecules are still present. So this ultraviolet absorbance will also reflect that how much of this contamination is loaded. So with a pure sample of DNA, the ratio of the absorbance at 260 and 280 nanometer is 1.8. So the instruments has the softwares which actually takes the absorbance at both these values, finds out the ratio, checks whether it is 1.8 or not. So if it is more than 1.8 or less than 1.8, then it indicates that there is a contamination of the protein or RNA molecules, and it is how pure is what is the extent of the purity of the DNA sample we are talking about. So ratios of less than 1 point indicate that the preparation is contaminated either with the protein or with the phenol during the organic extraction technology, and if it is more than 1.8, it indicates the presence of the RNA molecules. So sometimes if it is too much high or too much less, then we still need to go back and give the repeat the purification step, either by if there is by doing a chloroform extraction once, or by adding proteinase or RNase and incubating it at ambient temperature, and then again precipitating the purified DNA and again checking the purity of the DNA by UV spectrophotometry. So it is a very, very important technique which tells us the concentration of the DNA as well as the purity of the isolated DNA. Apart from this, because as I said that the total cell DNA which we are isolating, we do not just isolate from the bacteria. Now bacteria is a very simple host and therefore we have seen very generalized strategy which is there. But you sometimes have to use the plant cells, or you have to use animal cells, you have to introduce the DNA into that, or you have to isolate their genomic DNA and look for certain genes of interest which we want to do. So other methods of total cell DNA preparation are also employed, such as one is the CTAB method. So in this CTAB method, it makes use of a detergent which is called as cetyl trimethyl ammonium bromide, which forms an insoluble complex with the nucleic acids. So as you can see, this CTAB method, during this CTAB method, this can be used for the isolation of DNA from gram-positive bacteria, because it is difficult to break open the gram-positive bacteria, or from some kind of other cells which are basically utilized, tissue cells, plant cells, all those cells can be used for this. CTAB method can be used. So CTAB will form a complex with the DNA, and as you can see, when you centrifuge it, the nucleic acids bound to the CTAB will just precipitate out, and then you can discard the supernatant and resuspend this CTAB into resuspend this CTAB and then remove the CTAB to get the purified DNA. So these treatments will remove the CTAB, but this is a very, very efficient method, and again give rise to a purified DNA, and might cancel the use of the ion exchange chromatography or such things also. It can directly give a very purified DNA which is there. So this was all about the isolation of the total cell DNA. So total cell DNA or the chromosomal DNA, genomic DNA, we need to isolate when we want to look at one part of that chromosome, one gene of that chromosome, and do the downstream applications. But we also need to prepare the plasmid DNA, generally for using as a cloning vector, because where we need to put in the gene of interest, or some of the genes of plasmid DNA we are interested in. So we always need to isolate the purified plasmid DNA. When we are isolating the total cell DNA, we are isolating the plasmid DNA, but along with the genomic DNA. So we now, when we want to purify this plasmid DNA, we need to separate it from the genomic DNA, and obviously we need some extra steps. So the basic generalized strategy for the preparation of total cell DNA remains the same. So there will be growth of bacterial culture, there will be harvesting of the cell culture, there will be lysis of the cells to release the contents, there will be the steps such as the purification of the plasmid DNA or precipitation of the concentration of the plasmid DNA. So generalized strategy remains the same. The extra step that it requires is, which is basically to separate the plasmid DNA from the large amount of chromosomal DNA now that is also present in the cells. Now we do not need the genomic DNA or the chromosomal DNA, but we only need the plasmid or purified plasmid DNA which is there. Now plasmids are also varying in sizes. When we talked about cloning vectors, we talked about the variety of plasmids which are there. So they can be small molecules such as for some 1000 base pairs or 2000 base pairs, and they can be as large as running in the megabases. So obviously the larger plasmid DNAs will be much more difficult to isolate, but the smaller DNA molecules can be isolated. And then we also talked about that there will be high copy number plasmids, there will be low copy number plasmids. So high copy number plasmids means more number of copies present per cell. So if there are more number of plasmid molecules which are present per cell, there will be a higher efficiency of the isolation of the plasmid DNA. So again, this we will talk about a very generalized method that how the chromosomal DNA and the plasmid DNA can be separated and plasmid DNA can be purified. But again, it will depend upon what plasmid, what bacterial species or what microbial species or what cell species we are talking about, what cells we are talking about, and therefore many modifications are available, and in fact more and more methods are being explored to increase this efficiency on a daily basis. So we will talk about a very generalized method out here. Now as I said, the separation can be done on two different aspects. The first one is the separation on the basis of size. Now genomic DNA or chromosomal DNA is generally larger. When we are using the plasmid vectors or when we are using smaller vectors which are plasmids, then they range in the size from 2 kb to some 4 kb, 5 kb, that kind of a thing. But genomic DNA is, chromosomal DNA is in the range of MBs. So it's very large. So we can actually distinguish between the genomic DNA and the plasmid DNA and separate them on the basis of size. And how do we do that is that if the cells are lysed under very carefully controlled conditions, when we are lysing the cells, when we are cutting open the cells and when we are isolating the DNA, there tends to be a mechanical shearing that can occur. If we do this lysis and if we do the isolation of the total cell DNA in a very precise and controlled manner, then what we tend to get is a very intact DNA. So we will get the total cell DNA in which the genomic or the chromosomal DNA will be there and the plasmid DNA will also be there, and both the DNAs will be highly intact. So if we are able to do that, if the cells are very much lysed under a very carefully controlled condition, there minimal amount of the chromosomal DNA breakage will occur. The resulting DNA figments are still very large, much larger than the plasmids, and therefore can be removed with the cell debris by simple centrifugation. So this is a basic mechanism. Another factor that aids in that is that the bacterial chromosome is physically attached to the cell envelope. So because the bacterial chromosome is physically attached, if we do not do lot of mechanical shearing and we very carefully lyse the cell, very slowly lyse the cell, in fact the spiroplasts are formed so that there are smaller vesicles which are there, the bacterial chromosome will just come out very intactly and it will remain attached to the cell envelope. So when we centrifuge it, then the heavier part tends to settle down and therefore the bacterial chromosome genomic DNA will settle down, and in the supernatant what you get is only the plasmid DNA that you can again precipitate and concentrate to get the purified plasmid DNA. So this is like when it has to be done in a very controlled manner, and when you know that the plasmids are smaller in size. As I said, there are megaplasmids which are there. They cannot be isolated by this kind of a simple technique because they will be of the size of the genomic DNA. So you cannot separate by simple centrifugation that they will remain in the supernatant and the higher thing will settle down. It will not happen. But this can be done if you already know that your plasmid is very small and you are doing the lysis and the release of total cell DNA in a very controlled manner so that the chromosomal DNA almost remain intact and you just separate it by simple centrifugation. So this is how basically, when you do a very controlled lysis, so you can use certain special enzymes which are there. So in presence of lysozyme and edita, if you add sucrose to it, so now sucrose will make the cell wall labile and will tend to form such kind of structures. And if you add the precipitation, for the further lysis, if you add Triton X100, then it will just cause the total cell DNA to come out intact. So such, if you change the different chemicals or if you add on different chemicals for this isolation, then probably you will get intact chromosomal DNA, and that to adhere to the cell wall, the fragments of the cell wall. Therefore, when you centrifuge it, they will tend to settle down with the cell debris, and what you get on the higher supernatant is a intact plasmid molecule that you can take out. So again, I have told you the disadvantage is if the plasmid sizes are large. And for that method, we use another aspect of the plasmids to separate out from the chromosomal DNA, and that is that we do the separation on the basis of conformation. Now, conformation is the structural organization of the plasmid DNA. Now, you can refer here one of the lecture in molecular biology on topology of the DNA. So, in topology of the DNA, I have talked in detail about the various conformations of DNA inside a cell. Just to recall, I will just take over the simplest one, that the double-stranded DNA circles can take up one of the two quite distinct configurations. One is termed as the supercoiled DNA. So, as you can see the supercoiled DNA, both the strands are intact, and the circular DNA molecule is again taking this kind of it's basically supercoiled onto itself. Now, generally in a resting cell, the DNA will be in a supercoiled form. It will not be in a relaxed form, and that has molecular biological significances, because when it will come into a relaxed form, then it will release energy. Because the cell is keeping it under a constraint, it is storing the energy. When this constraint is removed, it will release the energy, and that will cause the disruption of the hydrogen bonds between the two strands of DNA, and it will tend to open up. So, for the processes of transcription, for the processes of replication to occur, it is favorable that the energy is produced by the uncoiling of the DNA, and there are special enzymes that will bring this about, and they are called as the topoisomerases. So, topoisomerases will cause the supercoiling to go from the supercoiled to the relaxed state, and then relaxed to the supercoiled state, so that the disruption can be used and the energy can be used. So, you can look into the details in the topology of DNA lecture, which is also present at the side. So, another configuration is the open circular thing. So, this is also called as the closed circular molecule, and this is called as the open circular molecule because when there is a nick in one of the strand, the phosphodiester bond is broken. So, when there is a nick on one of the strands, then the supercoiled form will become relaxed. So, you can see that the double stranded DNA circles can take up two different forms. Now, obviously structurally these two forms are different. The size of these two forms will also differ. So, because of the difference in the conformation of the genomic DNA and the plasmid DNA, we will try to separate it out. So, let us look at those techniques. So, an important technique involved in that is what is called as alkaline denaturation. So, the basis of this technique is that there is a very narrow pH range at which the non-supercoiled DNA is denatured, whereas the supercoiled plasmids are not. So, when we are talking about plasmid DNA, then the smaller plasmid DNA molecules generally in a resting cell occurred in the supercoiled form, or they occur as this open circular form, but the chromosomal DNA is in a linear form. It's a very big molecule, it's a linear molecule, and it is lying in a very different conformation than these two conformations. So, these two conformations or this particular conformation is the plasmid conformation. So, what we do is when we do so this is related to a very open circular, and if we create a cut here, it will become linear. So, chromosomal DNA is more relaxed than the plasmid DNA, which is in a supercoiled form. Now, altering the pH of the solution, we can see that this form will denature or in fact the larger linear forms or the non-supercoiled form will denature at that very narrow range, whereas there will be no effect on the supercoiled form. So that is what is used by this alkaline denaturation method. So what we do is that, for example, this is the total cell DNA. Now total cell DNA, this is a schematic representation. So this total cell DNA contains the plasmid DNA as well as the linear genomic DNA or the chromosomal DNA. So when you increase the pH, when you increase the pH, you make it alkaline, you raise it to the 12 to 15 range, then as you can see, the linear DNA will denature. So linear DNA will denature, however the supercoiled form will not denature, and whatever little amount of denaturation is also occurred, when you just lower the pH again, then they will snap back. The complementarity of the two strands of DNA, so they tend to snap back to join together. Now, even if the supercoiled DNA or some open circular DNA has denatured a little bit, as soon as you lower the pH by addition of say potassium acetate or something, as soon as you lower the pH, then they will snap back and regain its original form. However, the large linear DNA molecule will form a tangled mass. They would never be able to reanneal in a proper manner as they were in the resting cell or the intact cell. So when you increase the pH, make it alkaline, and then you decrease the pH, then this genomic DNA or the large linear chromosomal DNA tend to form a tangled mass, whereas the supercoiled DNA, they are intact molecules. So now this tangled mass becomes much heavier, and when you centrifuge it, it will separate out and become a pellet, and then you take this supernatant and put it into another tube and proceed with the purification or the basically concentration steps as involved in the total cell DNA. So this is one important method of alkaline denaturation and routinely used for the isolation of the plasmid DNA. Now we can also isolate manually by using making different chemicals, and nowadays companies also provide various kits that contain formulation of these chemicals step by step, and they just tell you keep adding, and then they have a spin column at the end, and at that spin column, when you spin, the DNA, the purified DNA will be on the membrane, and then after that you elute it into. So it becomes much more purified and a very a quick and faster method, and the chemicals are all prepared and come in a formulation in a kind of a kit which are readily available. But we can always do it by ourselves by preparing the different solutions and concentrating and isolating and purifying the DNA by the methods which I am telling. So an additional advantage of this procedure, when we talk about this alkaline denaturation method, an additional advantage of this procedure is that under some circumstances, specifically cell lysis by SDS, and as I said, neutralization with sodium acetate, most of the protein and RNA also becomes insoluble and can be removed by the centrifugation step. So further purification by organic extraction or column exchange chromatography may not be required. So alkaline denaturation is a very quick method to isolate the plasmid DNA. You just you lyse the cell, you grow the cells, you harvest the cells, you lyse the cells, and in the alkaline. So SDS is used in the alkaline in the presence of sodium hydroxide. So you give an alkaline environment, you lyse the cells, and then you neutralize it. Again, decrease the pH by adding sodium acetate, and therefore most of the protein, RNA, cell debris, genomic DNA, chromosomal DNA, all will be basically pelleted out, that you can precipitate, take the supernatant containing the plasmid DNA and then precipitate the plasmid DNA in a similar manner by ethanol precipitation. Another method which is used for highly purified and large quantity of plasmid DNA is what is called as ethidium bromide cesium chloride density gradient centrifugation. So this is based upon density gradient centrifugation. So what happens in the density gradient centrifugation is that when you centrifuge, then the compound which you have added into a column, there will be a gradient that will be formed. So in this case, the cesium chloride will be used, and the cesium chloride, if you use, there will be a gradient of the different concentration based upon the specific gravity. The cesium chloride will kind of form a different gradient, and when you use it as for the centrifuge with the biomolecule, then the biomolecule which is equating that particular cesium chloride value, it will lie at that point. So this is kind of a gradient. So the heavier molecules, the lighter molecules, and based upon the specific gravity, they will occupy different places. So they will be kind of separated and purified. As you can see, if a cesium chloride gradient formation is there, then the this DNA is comes at around 170 microgram per centimeter cube. However, RNA is settling down here. So this is the cesium chloride density gradient centrifugation which is used here. Now this is for the purification of DNA. It can be used for the purification of DNA from RNA or other protein molecules. Now within the DNA, if we need to separate the supercoiled and the linear DNA, what we use is called as the ethidium bromide. So ethidium bromide is a dye, it's which basically binds between the double helix, and within the double helix it kinds of create a little it changes the structure of the DNA molecules. So ethidium bromide DNA molecule, the double helix intercalation will cause the difference in the linear DNA as well as the supercoiled DNA. So it will basically decrease the specific gravity of these two molecules to different extents. So addition of the ethidium bromide will cause the supercoiled DNA, which is the plasmid DNA, and linear DNA to come at different levels in the cesium chloride column. Therefore, this ethidium bromide column can be extracted, ethidium bromide can be removed by extraction, and then finally cesium chloride molecules can also be removed and purified plasmid DNA can be obtained. So we have talked about the isolation of the total cell DNA today and the isolation of plasmid DNA today. We will continue with the purification of DNA, talking about the bacteriophage DNA isolation in our next series. Thank you.